Swastiastu. Good morning, sir, and good morning, everyone. Today, I want to present about photosynthesis. Before that, let me introduce myself. My name is Putumeda Chandra Cahyani with last name 1011. The first question about concept of photosynthesis. Explain the definition of photosynthesis and the main purpose of this process of plants. Photosynthesis is a biochemical process in green plants that converts light energy into a chemical energy in the form of glucose using CO2 and H2O with the help of chlorophyll. This process occurs mainly in the chloroplast. The main purpose of photosynthesis is to produce food or glucose as a primary energy source for plant metabolism and growth, as well as to produce oxygen as a byproduct, which is important for other living organisms. The next question about like reaction. Explain the mechanism of the like reactions, including the location in the chloroplast, the role of photosynthesis 1 and 2, and the product produced. Like reaction occurs in the thylakoid membrane of the chloroplast where chlorophyll absorbs light energy. Photosystem 2 absorbs light and splits water or photolysis to produce O2, electron, and H plus ion. The electron then flow through the electron transport chain, generating ATP through phosphorylation, and photosystem 1 further energizes the electrons to produce an ADPH. Therefore, the main products of the like reaction are ATP and ADPH and O2, which are essential for the next stage of photosynthesis. The next question about role of light. Why does high light intensity increase the production of ATP and NADPH? Explain conceptually. High light intensity increases the amount of energy absorbed by chlorophyll molecules, which enhances the excitation of electrons in the photosynthesis. As a result, the rate of electron transport increases, leading to higher production of ATP and NADPH. However, this increase only continues up to a certain limit before other factors become limiting. Next question about dark reaction or Calvin cycle. Explain the main stage in the Calvin cycle, including carbon fixation, reduction, and regeneration of REBP. The Calvin cycle takes place in the stroma of the chloroplast and consists of three main stages. First, carbon fixation occurs when CO2 is attached to RUBP with the help of the enzyme rubisco. Second, in the reduction phase, ATP and NADPH are used to convert the fixed carbon into G3P. And the last or finally, in the regeneration stage, some of the G3P molecules are used to regenerate RUBP so the cycle can continue. Next question, case analysis. Why under certain conditions does an increase in ATP and NADPH not lead to an increase in glucose production? This happens because the dark reactions are not only depend on ATP or NADPH, but also on other factors such as CO2 availability and enzyme activity. If CO2 is limited or enzymes like Rubisco are not functioning efficiently, the Calvin cycle cannot process optimally, resulting in low glucose production even when ATP and NADPH are abundant. Next question. The relationship between light and dark reaction. Analyze the relationship between the light reaction and the dark reaction in the process of photosynthesis. Light reaction and dark reaction are closely interconnected. Light reaction provide ATP and NADPH as energy and reducing power for the Calvin cycle. In turn, the dark reaction use these molecules to fix CO2 into glucose. Without the products of the light reaction, The Calvin cycle cannot occur efficiently, making both processes interdependent. Next question about limiting factors of photosynthesis. Explain the factors that can limit the rate of photosynthesis other than light. Several factors can limit photosynthesis, including CO2 concentration, temperature, water availability, enzyme activity, and chlorophyll contact. For example, low CO2 levels reduce carbon fixation, while extreme temperatures can affect enzyme function, ultimately slowing down the overall process. Next question about role of the enzyme rubisco. Compare the energy contribution of NADH and FADH2. Rubisco is the key enzyme responsible for fixing CO2 during the Calvin cycle. It catalyzes the reaction between CO2 and RUBP. The efficiency of this enzyme greatly affects glucose production. If Rubisco works efficiently, more CO2 is fixed into organic molecules. But if its activity is reduced or if photorespiration occurs, glucose production decreases. The next question, photosynthesis disorders. If there is a lack of CO2 in the environment, what is the impact of on the dark reaction and glucose production? A lack of CO2 directly affects the dark reaction because CO2 is the main substrate for carbon fixation. Without sufficient CO2, the Calvin cycle slows down or stops, leading to a significant decrease in glucose production even if like reactions are functioning normally. Next question, or the last question about metabolic integration. Explain the relationship between the product of photosynthesis or glucose and cellular respiration in plants. Glucose produced during photosynthesis is used in cellular respiration to generate ATP, which is required for various cellular activities. During respiration, glucose is broken down into CO2 and H2O, which can then be reused in photosynthesis. This creates a continuous cycle that links energy production and utilization in plants. Okay, that's all for my presentation. Thank you.